Well, welcome to this lecture on chapter 1 of the Lore of Running by Tim Noakes. In this chapter, Dr. Noakes goes through muscle structure, and I'll try to point out some of the reasons why he does this, but it goes to really understanding performance. We need to understand the parts that make up the human to understand human performance. Unfortunately, this is a little bit of dry material, so there's no great way to go through this. You should have already had a class on anatomical kinesiology, so you're exposed to some of this already. Also, your bio or KIN 223, 224 will serve you well for reading this chapter, and even KIN 391, you've done some of this. So I'm going to go through this somewhat quickly to sort of get through some of this material in order to get to some of the performance issues that we're interested in. All right, so let's start with muscle classification. There's different types of muscles classified based upon their shape. So there's smooth muscles. It's under the control of the autonomous system, spindle shape, uninucleated, and then there's striated muscle, cardiac in the heart, and skeletal. Obviously, we're interested in the skeletal muscle for the most part. Ultimately, we may be interested in smooth muscle if we're interested in how the GI tract is performing or responding to exercise, or really if you're having issues with endurance exercise and GI problems. But for right now, we're going to focus in on the striated and specifically the skeletal muscle, which is this bottom picture here, striped organization of muscle. Human body contains over 400 skeletal muscles, and it's 40 to 50% of your total body weight, typically, depending on your percent body composition. That would be your fat and your muscles and then all other parts, depending on which compartment model you're using to describe the human. Characteristics of muscle: it's irritable tissue. That means that the muscle can respond to a stimulus, electrical stimulus, typically. It has the characteristic of contractility. That means it can shorten. It also has the ability of extensibility, which means that it can lengthen, not under force, but it can lengthen. And elasticity, which is the ability to return to a resting length. So you can stretch a muscle, you can release it, and it will go back to its resting length. So you can extend the muscle and release it. The muscle will contract. And really, that's all it can do, is try to contract or resist being extended. The structures in a typical cell that you would have learned in anatomy and physiology: nucleus, plasma, endoplasmic reticulum, microfilaments. All of these typical cells are in a muscle cell. We also call a muscle cell muscle fiber. So when you hear the term muscle fiber, just realize you're talking about a typical cell. It's just that the structures in the cell are organized in a way that allows for the muscle to contract. So the growth structure of a muscle, this is just a picture out of a textbook. I'm just going to point out some of these structures. Epimysium is connective tissue that surrounds the entire muscle. And it's right here on the picture, this area here. Perimysium in here surrounds bundles of muscle fibers. So remember, that means bundles of muscle cells. These are called fascicles. Okay, so this is a fascicle right here, a fascicle being pulled out from within the muscle in terms of the drawing. And then endomysium, the connective tissue in here that surrounds individual muscle fibers. So this is all connective tissue. And that's important because Noakes will talk about the importance of connective tissue when we start looking at human performance. And connective tissue is not just the tendon that we talk about or ligaments, but also the epimysium, the perimysium, and the endomysium. And then ultimately we get down to sarcolemma or cell wall. And that's the trick. We just put sarco or myo in front of a lot of the typical cell structures. And that means it's pertains specifically to a muscle organization. So sarcolemma, cell wall, here's a nucleus. Sarcoplasmic reticulum, just like endoplasmic reticulum. Myofibrils, just like microfibrils in a typical cell. And we typically would say myofilaments here, microfilaments in a typical cell. All right, this is just another drawing of this, and this is just looking at a side view of a muscle cell, a muscle fiber. And the filaments in the cell are organized in a specific way that they overlap with each other, and ultimately they are able to interact with each other such that they can shorten the muscle fiber and create a pulling force on the ends of the muscle. All right, and we'll go through that. And typically we'll look at the striated pattern here, and that is due to the overlapping regions of the filaments, the myofilaments, microfilaments in a typical cell, the myofilaments within a cell. We have some distinct organization of the muscle in such that you can identify the stripes, the striated pattern here. And we call this section here the Z line and the Z line. And the contents between two Z lines is referred to as a sarcomere. This is the functional unit of a muscle cell, okay? This whole thing is the muscle cell. The sarcomere is just one unit within the muscle cell, and that's important to know. So here's the interaction of the myofilaments within the sarcomere, the myosin, or the thick filament, and actin, the thin filament. So in this cartoon, the red is the myosin, and the blue is the actin. And the myosin are thick units that have these protrusions that reach out to the thin filaments. Remember, this is 3 dimensions, okay? This drawing is just showing 2 dimensions. And those filaments will interact in a way that the sarcomere can shorten. And as such, the band will have different lengths based upon the shortening. Okay, so here's some sarcolemma, muscle cell membrane, myofibrils, thread-like strands within a muscle fiber, actin, myosin, sarcomere, the functional unit of the muscle cell. The Z line is the anchoring of the thick filaments. The M line right here is the middle of the sarcomere. That's where the myosins are anchored. And H zone. The H zone is right here in the middle where there's no overlap. And only one type of, and I think my next picture has a better picture of that. The A bands are darker, and they represent where there is overlap between the thin and the thick filaments, and the I band is no overlap, or this region here in this part of the drawing. All right, this is out of your textbook, figure 1.3. And in this part of the textbook, Noakes really emphasizes the identifying elastic tissues. And remember, elastic tissue is protein, collagen. And he identifies some elastic tissue within the sarcomere, titin, which extends from the Z disc to the M line. So here's the Z line. And the elastic region of PEVK, that's this part right here, the elastic portion of titin right here. And this whole thing is the titin, and this is the non-elastic portion of titin. So it goes from the M line to the Z disc. And PEVK just represents different amino acids that make up this elastic region. And type 2 fibers tend to have more titin than type 1. Elastic tissue there are some advantages to it. We'll talk about it a bit. In fact, in this chapter, Noakes does raise the issue of whether or not connective tissue, the amount of it, or the damage to it, is related to fatigue. Now, we got to get into what fatigue is, but right now I'll just say fatigue, I'll use a general definition, is the reduced ability to generate force. And here I mean muscle tendon force, so the muscle tendon unit together as opposed to a muscle cell by itself. And so if there's damage to the connective tissue, there could be a decrement in the ability to generate a pulling force on the bone via the tendon. And maybe that has something to do with hitting the wall. We don't know. That's one hypothesis. Okay, some more cell structure. Within the sarcoplasm, and this is important to recognize, there's a sarcoplasmic reticulum, endoplasmic reticulum in a typical cell. This sarcoplasmic reticulum is the storage of calcium. And down the road, we're going to be talking about hydration. We're going to be talking about types of things to put in drink. Calcium is one of those. And we'll talk about whether or not that should be replaced. And then there's transverse tubules. Those are the cell wall sort of goes down inside the cell here and interacts with the sarcoplasmic reticulum. And we'll see in just a bit, when a muscle is told to contract, an electrical signal will go down within the transverse tubule, and it will indicate to this the terminal cisterns to release the calcium from within the sarcoplasmic reticulum, and that calcium is important for muscle contraction to happen. All right, this is just a neuromuscular junction. This is where the motor nerve or motor neuron meets the muscle fiber or the muscle cell wall. It's a motor end plate. It's a pocket formed around the motor neuron by the sarcolemma, and the neuromuscular cleft is a short gap between. So this gap between the motor neuron and this whole cell wall, this is the sarcolemma, this blue part here. When a signal, a motor signal comes down the motor neuron, it causes a release of acetylcholine that jumps this gap and causes a depolarization on the cell wall of the muscle cell, and then that motor signal continues to go along the cell wall. And as I mentioned in the previous slide, it goes down inside the T-tubule, ultimately causing release of calcium. All right, some more key components within the muscle cell. Mitochondria, these are the powerhouse. This is where aerobic conversion of chemical energy is transformed to ATP, and that ATP is then used by the muscle to generate muscle force. So it really is the source of transforming chemical energy to mechanical energy. So it's within the muscle cell and the cytoplasm, or really sarcoplasm would be the correct way to say that. It's the location of oxidative conversion of foodstuffs into usable cellular energy. Oxidative conversion just means use of oxygen to transform chemical energy to something else. So oxidative conversion. And is the number of mitochondria, is the mitochondrial density related to endurance performance, and is it a limiting factor in endurance performance? It's a good question, and something we'll talk about as we go along. We need this for aerobic exercise. This is ultimately going to be related to VO2 and VO2 max, rate of oxygen consumption. Because without mitochondria, you aren't able to use oxygen to convert chemical energy, carbohydrates, fats, over to ATP that's then used for adenosine triphosphate, which is then used for muscle contraction. So this is just some elements within the muscle. Figure 1.2 in your textbook, sarcomere, functional unit within the muscle cell. This whole thing is the muscle cell here. And you can see the A band, the dark band, this light region being the I band. Here's the I band. And I'll talk about this in just a bit here. So those are the mitochondria identified. Within the muscle cell, there's also fat droplets. These things right here, TG, triglycerides. Noakes will talk a lot about triglycerides throughout the textbook. So you're going to want to get familiar with what a triglyceride is. It's 3 molecules of fatty acids and 1 molecule of glycerol. These can be used for fuel. These can be used as potential chemical energy to convert to ATP, adenosine triphosphate, ultimately used for generating muscle force. So it's a source of energy. Lipolysis, the enzyme that you need for that is lipase. So fat can be a big source of fuel, and we're going to talk about that. Glycogen is another component within a muscle cell, right here and right here. It's a polysaccharide, i.e. that is an animal starch. That means just lots of glucose molecules, lots of glucose. And glucose can be used in the mitochondria for aerobic conversion of chemical energy to ultimately generate ATP. And it can also be used outside the mitochondria, and we'll talk about that, in anaerobic, not using oxygen to generate ATP. Glycogenolysis is the breakdown of glycogen to glucose. That's a good term to know. Glyconeogenesis, formation of glycogen from non-carbohydrate sources. So it's new glycose, neo. Glycogenesis, formation of glycogen from carbohydrate sources. So these are 3 terms that you're going to want to know, especially for a test situation. Good to be able to distinguish between these. Glycogen is stored in all muscles, and it's also in the liver. And this is something that we're going to talk about quite a bit, is the amount of glycogen in the liver, the amount of glycogen in the muscle, ready to be used for ATP, and the transport of glycogen from the liver to the muscle via blood. And so we're talking about blood glucose as well. The total glycogen storage in the muscle is relatively small, but we'll talk about that. We'll talk about carbohydrate loading, why you may try to do that before an endurance event. And it can be depleted in a few hours of exercise, maybe 2 hours, maybe even shorter, depending on how well you're trained or how well you're fueled. So if you're trying to do, already you should be thinking, if you're trying to do a long endurance event, 10 hours, 12 hours, 16, and if you run out of muscle glycogen, you've got to be able to have some other fuel. That might be triglycerides, might be delivered from the liver, or what have you. And we'll talk about that. All right, fiber typing. I actually don't do a lot with fiber typing, but it is good to distinguish between different types of fibers. Some fibers are more suited for aerobic conversion of glucose to ATP, and some fibers are more suited for anaerobic, not using oxygen to convert glucose to ATP. And so we typically break them into 2 main categories, type 1 and type 2, but there's all sorts of subtypes within type 2 especially. And there's still lots of discussion about the fiber typing within the muscle cell. So I typically just will, throughout the semester, really focus in on type 1 and type 2 as a general category and more as a concept of training and which type of cell you're trying to train, and making sure that you're doing the right, appropriate, the correct stress to stress the system in order to get a training response. Here's just a little table summarizing some of the key things. Number of mitochondria, high in type 1 because lots of aerobic conversion of glucose. Low in type 2, maybe some intermediate, a little bit of both. Resistance to fatigue, type 2 is lower because these are anaerobic, where you can only go for so long. But type 1, lots of resistance to fatigue. You can use these for long durations, or postural muscles standing for long times is largely done by slow twitch type 1 fibers. Predominant energy system, anaerobic, maybe a combination here, aerobic here. ATPase, this is just the enzyme needed to get some of the energy out of ATP. The speed of shortening is faster for type 2 than type 1. Efficiency, I don't really like this term, but we do get to use it in terms of how much energy you get out of a system versus how much energy you start in a system. Type 2 tend to be low in terms of efficiency. Think about it this way, is that when you do high intensity sprints, you get hot faster than maybe doing a low intensity, long duration run. That buildup of heat quicker in the sprints is because there's less efficiency. You're generating more heat for a given level of energy input. And the tension that you can generate tends to be higher in type 2 and moderate or lower in type 1. But a lot of our muscles are a mixture of these, and so sometimes it's hard to distinguish between them. And we'll actually talk about this later on in terms of training, is that you might actually, as an endurance athlete, really benefit by having and training some of the fast twitch fibers. And that's why a lot of endurance athletes nowadays are including strength training within their endurance training program. So we'll get to that. All right, so quick summary: muscle structure. You should know your gross anatomy down to your cell level, classification of muscle cells, or excuse me, of types of cells, characteristics of a muscle, muscle structures, the connective tissue issue, key components within a muscle cell, and a little bit about muscle fiber. All right, that's this part of chapter 1 of Tim Noakes' book.